Okay, so moving on with the next question. So this one is asking you to state how you are going to calculate the percentage change in volume of the red tissue in each cube. So what you can actually do is, for the first part of the question, you can actually state the formula. So the formula would be the initial volume of the cube, you minus that with the final volume of the cube, and then you are going to divide by the initial volume. And of course, now we need to remember that it's talking about percentage, right? So we need to make sure that we include in the formula, we times that with 100. So just description of how we calculate the percentage change. Okay. So in the next question, we have another experiment that's being done by the student looking at the effects of temperature on the release of betadine pigments from the beetroot tissue. So they mentioned the procedures here. They cut the beetroot, okay? They prepare beakers with water. The beakers were then placed in water baths at temperatures between 0 to 85 degrees Celsius. The beakers were allowed to equilibrate for 5 minutes. The cubes were added to the beakers that's prepared, and after 30 minutes the beetroot cubes were removed. We use the colorimeter, each with a blue-green filter, to measure the percentage transmission of light through the fluid remaining in each beaker. Alright, and we have the table. The result of this experiment are shown in table 1.1, right? So the first bit here, so we have the temperature. Okay, we have the percentage transmission of light, of how much light can actually pass through the beaker, okay, once the pigment has been released. The student decided that two of their results are anomalous, which is the one that does not fit the rest of the values. Okay, what? So any anomalous result is where you can see that, oh, it doesn't really fit the pattern. All right. And one of the results that are considered anomalous has been circled, which is here, 82. Circle the other anomalous result and complete the table by calculating the mean percentage transmission of light at 25 degrees Celsius. And of course, what you need to do is that we look at the data, and here we can identify that at 65 degrees Celsius, okay, the anomalous result is from student number 4. Okay, so we circle that, and then we proceed with calculating the mean. So here, like with any other formulas, I'm sure you know how to calculate the mean. What you need to do is you total up all of these values. Alright? Oh, oops. So you total up all of those values and you just divide it by 4. Okay. So I think you should be able to do that. So the answer should be 89.3. Okay. Oops, it's a bit too thick. I'm just going to use that one. Okay. Describe 2 ways in which the method described in C could have been improved to give better repeatability between the results of the different students. So meaning, what information are we missing in the methodology, and what are the things that should have been standardized or should be kept constant. So if you look at the bits of the methodology that is mentioned here, all right, okay, number 1, okay, what could be the problem with here? So we can see that they mentioned that the cubes of beetroot were being cut, but we know that, okay, there's no information how it's being cut. So, all right, maybe the idea of how we should have been cut. Okay, so when we want to cut, okay, what are the bits that we need to be standardizing? So make sure that when they are cut, all of the cubes are of the same size, right? Cutting them at the same size. Oops. Sizes or even volumes, okay? Right? Okay. Or even better, if you can remember the idea of surface area to volume ratio. Okay. Now, the next thing is, okay, maybe we can also talk about the beetroot itself, right? What about the beetroots that should be standardized or should be kept the same? Okay. Other points that we could also include maybe in relation to the beetroot. Okay. And I put here in the notes, okay, the idea that we use the same age, same species, right? The next bit, okay, we have the beakers containing water were prepared. Now, again, we have to standardize the experiment, so we are able to repeat this experiment, okay, to increase its repeatability. So we have to mention or look at the description of the procedures that allow us to again do the procedures well. So what else should we be standardizing here? So they mentioned the idea of adding water to the beaker, but they didn't mention how much water. So perhaps what we can improve from here is mentioning, okay, standardizing or using the same volume of water. Okay, you can use the same volume of water. All right. Okay, when we use the same volume of water, when we're deciding what volume to use, you need to make sure that whatever volume is used has to be ensuring that, okay, enough to cover the cubes completely. Cover or submerge. And of course, that will be depending on how you would be cutting the beetroot in the first place. All right. And then we have the beakers placed in water bath. That's good. Okay. When we have when we have the water baths, okay, I also normally prefer the students to also make it into a good habit of, rather than just using a manually controlled water bath, like you just check with your thermometer. Okay, we have a better one yet, which is basically perhaps maybe use a thermostatically controlled water bath. Okay. All right. So when we're adding the beetroot cubes, okay, just make sure maybe perhaps ensuring that they are submerged completely. We allow the removal at the same time, so that's fine. They have standardized the timing here of how long you submerge the the cubes. And then we meant that they did actually mention here that they're using colorimeters. So when we use any sort of apparatus or machineries, we need to ensure that we're using the same colorimeters as well, because they're not what normally happens is different colorimeters might have a different sort of readings as well. So using the same colorimeters to for each temperature would be a good idea as well. Okay, now the next bit is probably what we can do is, because we're talking about having these pigments to that's going to be entering this beaker of water, right? So what we can do is we can uniformize, okay? We can make sure that this pigment are well mixed into the water before we actually read, okay? And put them into the colorimeters before we take any reading. So that would be a good example to add as well, to ensure that maybe, all right, stir so we stir before taking any readings. Now, whenever we want to take multiple readings, because we're going to be investigating them at different temperatures, and another important point that we should also remember is that remember to calibrate before each reading, okay? Okay, so those are the few bits that you can and may want to include into your answer here. All right, so I don't think I'm gonna rewrite those here, but yeah, whatever the points I've mentioned there, you can just transfer them here. That'd be good enough already, any 2 points. Okay, with reference to the table 1.1, state and explain the effects that increasing temperature has on the release of methane from beetroot tissue. So let's look at the table. What can we see? What sort of pattern that we can see? So as we go here, looking at the pattern, so as we go down, okay, as we increase the temperature, right, we can see that the transmission absorption, okay, you can see that the transmission of light reduces. So what does that even mean? So when there's not much light can pass through the fluid, that means that there's going to be more pigment. That means it's going to be darker. So more pigment has been released, okay, resulting in a much darker solution in the beaker. So if it's darker, so lesser light can pass through, so therefore lower transmission. Okay, so from that we can actually already write down here that stating, so they state, okay, is that increasing temperature increases bigger. So what we need to explain is what part of the plant or beetroot tissue here is being affected. So when we're increasing temperature, what we're most likely affecting is that we're affecting the membrane fluidity. Okay, why? Because our membranes, okay, the phospholipid membranes, the phospholipid bilayer, they actually have proteins as well. Okay, higher temperature or increasing temperature, okay, okay, and if it's too high, it actually causes the denaturation of the proteins. So when there is not much protein available, so what will happen, okay, is you're causing the permeability to increase. Therefore, more pigments passes through. Alright, there you go. And of course, if you want to go back and revise on this, okay, you can look at, okay, topic 4. Okay, when we look at the properties of the cell surface membrane. Okay. Alright, so that's the end of question 1. Okay, I'm going to stop that and then please check part 3 for question number 2.